Forget the DNA. Forget the mitochondria. Right now, inside every single cell of your body, a tiny, headless molecular machine is running the most important program in the universe. It is not alive in the way cells are. It does not think or decide. Yet it is building you, molecule by molecule. This machine is the ribosome, the bridge that turns genetic information into physical reality. Without ribosomes, genes would remain silent text, and life would have no structure, no chemistry, and no motion. Protein synthesis begins before the ribosome even starts working. Inside the nucleus of eukaryotic cells, a specific segment of DNA is copied into a temporary working message called messenger RNA, or mRNA. This molecule carries the instructions written in a 4-letter chemical language. Once formed, the mRNA leaves the nucleus and enters the cytoplasm, where ribosomes are already waiting. The ribosome does not interpret meaning. It simply reads what is written, with absolute precision. A ribosome is made of 2 parts, a small subunit and a large subunit. The small subunit binds first to the mRNA and moves along it until it finds the correct start signal. Only then does the large subunit attach. At this moment, the ribosome becomes a complete translation machine, ready to convert genetic code into protein. This conversion process is called translation, the transformation of RNA language into the amino acid language of life. The ribosome reads the mRNA 3 letters at a time. Each 3-letter unit is called a codon, and each codon specifies one particular amino acid. Nearby, transfer RNA molecules, tRNAs, are floating in the cytoplasm. Each tRNA carries one amino acid and has a matching 3-letter sequence that pairs with a codon. When the correct tRNA enters the ribosome, it locks perfectly into place, delivering its amino acid to the growing chain. This is the ribosome's real power. It is not just a reader of instructions. The ribosome itself catalyzes the chemical reaction that links amino acids together. It forms peptide bonds, stitching individual amino acids into a precise sequence. Codon by codon, bond by bond, the ribosome moves forward along the mRNA, assembling a protein with remarkable speed and accuracy. When the ribosome reaches a stop signal, the process ends. The completed protein chain is released, and the ribosome splits back into its 2 subunits, ready to begin again. The protein then folds into a specific 3-dimensional shape, sometimes assisted by other cellular systems. Only after folding does it become functional as an enzyme, a hormone, a structural component, or a signaling molecule. Some ribosomes float freely in the cytoplasm, producing proteins that function inside the cell. Others attach to a membrane system called the Rough Endoplasmic Reticulum, where they build proteins meant for secretion or for membranes. The ribosome itself does not change. Only the destination of the protein does. At its core, the ribosome is the point where evolution solved one of life's deepest problems: how to turn information into matter. This mechanism has been conserved for billions of years, from the earliest bacteria to modern humans. The ribosome does not think, decide, or adapt, yet through its relentless precision, it makes all complex life and biological evolution possible.